Now, I want to talk for a second about the different types of receptors we might see when we're talking about free-floating signals contacting receptors on the outside of membranes. So this wouldn't affect our signals from gap junctions, but it would affect most of our other types of signaling. So we said in the last video that the way cells communicate is that they're going to, in general, be releasing some type of signal that's going to activate a receptor protein in the cell membrane. But we have different types of proteins that can act as receptors, and they work a little differently when they're activated. So we're going to talk about 3 types. 2 of them are really more important for you to understand. And the first is called a G protein coupled receptor. Okay, so that's a long word, but we're going to go through it. So our receptor, in this case, our G protein coupled receptor, or GPCR, is an integral protein. So it's bound in on the membrane. It goes all the way through. So it would have, for example, a cup shape here on the outside of the cell that's going to bind to the signal. It would have a piece that goes through the membrane and then a bit sticking out somewhere on the inside of the cell. So when our signal binds in, it's going to bind in over here on the outside of the cell, and it's going to cause some type of change on the part of the receptor that is on the inside of the cell. And if we're talking about a G protein coupled receptor, that part of the receptor protein that's on the inside of the cell is going to do a really specific thing. When our G protein coupled receptor is activated, so after that signal binds in, what happens is that our G protein coupled receptor, so the protein that bound onto the signal, is then going to bind on to its other side, the intracellular part. It's going to bind into another protein, a peripheral protein, called a G protein. So steps, we have a signal bind on the outside, and then that receptor binds to a G protein. When it binds to that G protein, the G protein also gets activated. And what it looks like when it gets activated is it splits apart. Okay. The final step for this G protein coupled receptor is that that activated G protein is going to be the one that starts creating second messengers, that kicks off that pathway of dominoes inside of the cell, that does that signal transduction part of signaling. So that sounds like, I don't know, kind of extra steps from what we described in our previous video, and it is. So the reason we care about G protein coupled receptors is basically that you'll see in the future that these will often have kind of longer lasting effects. So they'll react to a signal for a longer time than other types of receptors. So there won't require that signaling molecule to be present all the time, because after that G protein is activated and creates a bunch of second messengers, that signal inside the cell kind of hangs around for an extra amount of time. This is different from our ligand-gated ion channel, which is our second type of receptor, also super important. So you should key into this word channel, right? We're talking about an ion channel. And we've talked about ion channels before. So when we're talking about channels, we talked about how they are membrane proteins that basically create a pore through the membrane that ions can travel through. So we talked about them when we were talking about passive transport, because we remember that since they just open up a tunnel through the cell membrane, that when they're open, all that's going to be able to happen, all that we're going to be able to do is to have molecules traveling from where they're high in concentration to where they're low in concentration, because it's just a hole. So channels were used for passive transport. Now, the ligand-gated ion channel is just a special type of channel And the thing that's special about it is that our ligand-gated ion channel, some of the time it's closed. It's a receptor protein because it can be inactive and closed. And the way we open up the channel, the way we create that tunnel, is that the signaling molecule binds on. So we have our inactive receptor here, our inactive ligand-gated ion channel. It's a protein that goes through the membrane. And when we have a signaling molecule, it binds on to the ligand-gated ion channel. So ligand actually just means like something that binds onto something else. So the signal here is the ligand. So our signaling molecule binds onto this ion channel, and that causes a shape change or conformational change in the receptor, which opens up the channel. So now the channel is open, it's activated. So this receptor is now activated, our channel is open, and because this channel is open, that's going to allow for passive transport. And specifically, it's going to allow for passive transport of ions. So these ions, these charged molecules or charged particles that aren't able to get through the cell membrane because they can't get through this double layer of nonpolar fatty acid tails, they're now able to diffuse across the membrane from high to low because we've opened up a channel. Our third type of receptor is something called an enzyme-linked receptor. We're not really going to focus on those, but you should know they exist. And what an enzyme-linked receptor is a receptor protein that when a signal binds to it, the part that's inside the cell, so the part of the receptor that's inside the cell, turns into an active enzyme. So you may or may not remember when we talked about proteins, we briefly mentioned that enzymes are a type of protein that act as a catalyst. So enzymes are just a protein that makes it easier for a chemical reaction to happen, that makes that reaction happen faster. So an enzyme-linked receptor, when a signal binds onto it on the outside, on the inside, our receptor protein becomes an enzyme, so that inside part starts speeding up some chemical reaction. Now, usually these enzyme-linked receptors are involved with something we call protein kinases, and they tack on phosphate groups to stuff, but we don't have to worry about that for the purposes of this course. And that's it.